Assalamualaikum and hi, we are from Group 3 and today we are going to present about lactic acid bacteria as pro-technological, bioprotective and health promoting cultures in the dairy food industry. So, before we start our presentation, let me introduce our group members, which is Nurul Aina Najwa binti Ruslan, Nur Aisyah binti Ayub, Nur Azana binti Azahar, and me, Nur Farhana binti Helmi.
Assalamualaikum and hi everyone, my name is Nurul Aina Najwa binti Ruslan. So today I will continue present about topic lactic acid bacteria as pro-technological, bioprotective and health promoting cultures in the dairy food industry. Lactic acid bacteria have long benefited humankind with an estimated history of safe usage in the food industry, mainly associated with their ability to enhance the storability of highly perishable raw materials through lactic fermentation. Lactic acid bacteria fermentation is widely reported as a biotechnological process to improve the overall quality and safety of fermented foods to different extent. According to the definition of microbial food culture, lab strains may be deliberately introduced into the food chains as pro-technological starters based on their probiotic potential or as bioprotective culture. Fermented dairy products are consumed around the world with enthusiasm and are appreciated across culture due to their nutritional value and their great appeal as traditional and ethnic foods. Okay, according to the specific psychochemical characteristics of different kinds of meat and of the technology employed for processing, fermented milk product can be categorized mainly as cheese, fermented dairy beverage, yogurt, kefir, buttermilk, acidophilus milk, and sour cream. During lactic acid fermentation, several biochemical modifications of food may be attributable to metabolism. In particular, desirable features of lab strains include technological properties such as fast acidification of the matrix, tolerance to food processing conditions, good proteolytic and lipolytic activities, and the biosynthesis of functional metabolites. During lactic acid fermentation, like protease, lipase, and B-galactosidase are involved in the enzymatic release of the volatile compounds responsible for the distinctive organoleptic and sensory profile of dairy products. Okay. Lab starter also culture must be designed carefully to ensure safety and preservation in artisanal foods to associated with the production of various organic acid, bacteriaceans, and other compounds with broad spectra of active microbial activity. That's all from me. Thank you.
Assalamualaikum and hi, my name is Nur Farhana binti Helmi and I will present about the process recombination DNA of this dairy product. So the first step is DNA isolation, which is DNA to be cloned or recombined. In this part, DNA containing DNA of interest. Then, the second process is restriction on enzymes. In this part, a desired DNA molecules from two different sources, which is I-lactate dehydrogenase chain and lactic acid bacteria cell, are treated with the restriction enzyme, which is restriction endonuclease. After that, the restriction enzyme will be dissected in the double strand of DNA at a specific short nucleotide sequence for both strands. Then, they producing staggered cut, which is sticky ends, and a knot with the complementary base pairs. So, the sequence with sticky end can bind to a complementary single-stranded tail or another to restore a double-stranded molecule. The next process is ligation enzymes. Ligation process by ligase forming phosphodiester bond of DNA backbone. Next is process exertion of DNA, which is transformed into a vector, which is host and will be expressed by the host in the large quantity. Lastly is process multiplication, which is dairy product will be produced after the recombinant process. So that's all from me. Thank you.
Assalamualaikum, my name is Nur Aisyah binti Ayub. I'm going to explain the comparison part. The pros and cons of GMO in dairy food industry. For the pros of GMO is a fewer pesticides, less cost and have more nutrients. For the cons may cause allergic reactions and may increase the antibiotic resistance. In this study, it can be said that the main common starter culture media used in dairy foods industry is lactic acid bacteria compared to the coagulase negative staphylococci, which is CNS. Okay, what are the difference between CNS and LAB used in dairy food industry? For coagulase negative staphylococci, CNS are mainly responsible for the development of the aroma as well as the flavor by their proteolytics and lipiotics activity. Furthermore, CNS are able to reduce nitric leading to the production of nitrosomoglobin, which is important for the characteristic red color of this fermented product. Super oxides dismutase and catalyst activity of CNS are important to prevent lipid oxidations by the detoxifications of superoxide radicals and the decompositions of hydrogen peroxides respectively. Meanwhile, for LLB represent a successful method for the biological controls of foodborne pathogens and food spoilage microorganisms in this product. Fermented dairy products have a less risk of contaminations as they are enriched in antimicrobial content, including ethanol, organic acids, and bacteriocins. The LLB in this product may inherently be present or they may be added by human interventions. LLB generate antimicrobials compound including organic acids, diacetyl H2O2 and bacteriocins during fermentations, and this product yields some advantage. These products of LLB metabolism yields advantage for the color, aroma, tastes, and texture of fermented foods while prolonging shelf life by inhibiting the growth of pathogenic and spoilage microorganisms. Therefore, LLB are of great importance for the dairy industry to improve the organoleptic quality of fermented meat products and to extend their shelf life. The organic acid generated by LLB induce antibacterial activity responsible for lowering the pH value. This is one of the key mechanisms in the quest to biopreserve fermented foods. LLB generate many organic acids including but not limited to acetic, lactic, succinic, formic, and propionic acids. Low pH makes organic acid soluble in all. Thus, organic acids pass through cell membranes. Assalamualaikum, my name is Nur Aisyah binti Ayub. I'm going to explain the comparison part. The pros and cons of GMO in dairy food industry. For the pros of GMO is a fewer pesticides, less cost, and have more nutrients. For the cons may cause.
Assalamualaikum and hi, my name is Nur binti Azhar. The profit of lactic acid by recombinant technology is risk of failure and contaminations can be avoided. For example, direct acidifications with lactic acid in dairy products such as cottage cheese is preferred fermentations. If we avoid these contaminations, we can avoid economic impact of foodborne disease that we can refer. The total productivity loss associated with foodborne disease is low, and middle income countries is estimated to cost US 95.2 billion per year, and the annual cost of treating foodborne illness is estimated at US 50 billion. Lactic acid bacteria perform an essential role in the prevention and production of wholesome foods. The lactic acid fermentation are generally inexpensive and often little or no heat is required in their operations, making them fuel efficiency as well. Lastly, adjust acidity and improve flavor, texture, and stability.